This Week in Virology, the podcast about viruses, the kind that make you sick. From ASM Microbe 2022 in Washington, D.C., this is TWiV, This Week in Virology, a special episode recorded on June 12th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in Washington, D.C. from This Week in Microbiology, Michael Schmidt.
Hello, everyone. I'm here to keep Vincent honest.
You know, Michael's wandering around, so I thought we should pull him up here and have a little
tete-a-tete.
We mix our talent among podcasts, right? Thank you, Michael, for joining us.
Thank you, Vincent.
We have two guests today who are science artists, and they're exhibiting right behind us today. And I thought you would like to hear about what they do and how they do it and what inspires them and so forth. These are two artists I have followed for years. I have purchased many of their items, and I'll tell you more about that later. But right next to me here from Trilobite Studios, Jane Hartman. Welcome to TWiV.
Thank you. It's so nice to be here.
You can see Jane is wearing glass virus earrings, so we'll have her explain all about those. And from Artologica, Michele Banks. Welcome back.
Thank you.
And you have been on TWiV before, right?
I have been on TWiV before, and I'm a painter. I live in D.C. All of my work is inspired by science. Well, we'll be talking about that.
Yeah, and you are here also in Rockville.
Yes, I'm in Rockville, Maryland, which is a suburb of D.C., so close by.
And so a couple of years ago, we had an American Society for Virology meeting at the University of Maryland College Park campus, and you came to that. So when there are local meetings, you try and get to show your works, right? That's great. So, Jane, Trilobite Studios, why did you come up with that name?
So my formal education was in geology and then paleontology for my master's degree. So Trilobite was a nod to my my education.
Okay, got it. That's good. And Artologica, where did that come from?
It's basically ars biologica, so Latin for biological art.
Good. I actually didn't know either of those stories, so that's good to know. Michael, have you owned any of their works?
I have to admit, I shamelessly own a tie.
After this, you need to go over and purchase some.
I need more ties.
You need more ties?
Well, you always need ties. You always yell at me when I wear a tie.
Well, I don't yell at you, but wearing a tie is pretty formal, right?
Well, I live in a formal situation at a medical school.
But you suggested yesterday to Michele she needs to make some bow ties. Then you'd really sell a lot of ties.
The infectious disease folks love bow ties.
Is that possible for you to make bow ties, Michele?
In theory, I can definitely make bow ties.
Okay.
I just wasn't sure if there was going to be a big market for them, but if you say the infectious disease guys like the bow ties,
you only need to look at Vincent's colleague, Dan Griffin, and watch This Week in Virology clinical updates. You're up to episode 100 and something or other.
Yeah, 118,
And Daniel wears a different bow tie for seemingly each episode. Yeah. And they're all microbiologically themed. Some are parasites, some are viruses, some are bacteria, and they're quite colorful. And Daniel can actually tie it without even looking. I mean, I saw him do that at the incubator as he was getting dressed for his, 2 minutes of fame for the photographers. He had his bow tie off, and he immediately put it back on. And he often has matching socks that go with the bow tie.
I feel like being able to tie your bow tie without even looking in the mirror is something that should go on your CV.
I guess I agreed. Do you own any bow ties, Michael?
I do indeed.
Does anyone wear a bow tie here in the audience? No. Okay. Michael, I'm not sure you're convincing her to get bow ties in. How about this? If you make bow ties and they don't sell, Michael will pay for them.
Oh, my. I don't know. Silk's expensive.
Okay. So let's start by describing your art for us. So, Jane, what kind of art do you make?
So I work in glass, and I use two techniques. The first one is called fused glass, which is made in a kiln. So I will cut out sheets of glass and piece it together and shape it and layer it, and then it goes in the kiln and it all melts together. So that's fused glass. I also do a traditional stained glass where you're cutting out pieces and fitting it together like a mosaic, and then you let it up and you solder it together. And so that kind of work is ideal in windows. And so in both cases, I concentrate on science themes, primarily biology, but I'll do any kind of science.
All right,
My
so
graduate mentor, Walt Konecka from Indiana University, his second career was stained glass, and he used the copper foil method to do stained glass, and he made some of the most beautiful things I have ever seen.
all right, so here's an example of your work. So this is
Yes.
fused.
This is fused glass. So I would start with a square of the green on the background,
Right.
and then I'm cutting out circles of various colors to form the envelope. That's Staphylococcus, and so I layered on top of the background, so it's like in some places it's quite thick before it goes in the kiln, and I put it in the kiln. And it'll go through a process of anywhere from 12 to 24 hours or more in the kiln, and it melts flat. Then it goes back in the kiln a second time to get that shape. So I have a mold in that shape, and I'll put the blank, it's called, into the mold and send it through the kiln again for another 12 or 24 hours to make that bent shape.
So there are little speckles here. How do you get that?
So I use what's called frit, which is basically crushed up glass, and
Okay.
I can make it myself, or I can purchase it already crushed up, and it comes in different sizes. So I'll
Okay.
use, coarse frit in certain places and fine frit in other places.
So you put
Yeah.
all that on, and then in the oven it all melts.
It all melts, right?
What's the temperature of that?
Typically, the top temperature is, excuse me, 1,500 degrees Fahrenheit.
Wow.
And getting it to that top temperature is tricky. There's a lot of science to it because you can't heat the glass too fast or it thermal shocks. Then you can't cool it too fast or it thermal shocks. And you also have to anneal it. So typically, at 1,500 degrees, the kiln cools to 900 degrees, and then I have to hold it there so the temperature all equals out among the glass so that it's all stable. Otherwise, it'll end up cracking. And sometimes a year or two later, it'll stress crack because if it's not annealed properly.
So it's the equivalent of doing something like making Damascus steel with the layering and the different temperatures and all of the other things that you're doing. You're really doing an artisan approach to it.
Yes, that's absolutely true. And so 1,500 degrees is a typical temperature, but if I wanted it to be less flat, if I wanted some contour on it, which a lot of my work has, then I might take it to only 1,400 degrees. So it's all sticking together, but you still get some dimension to it.
Now,
Yeah.
how did you learn how to do this? Are you self-taught, or did you?
I started mostly self-taught. I took a real quick beginning class to see, if I was real, whether it was I wanted to buy the kiln. And then I did a lot of reading, a lot of experimenting. And then as time went on, I started taking master's classes to learn different techniques. And
How big is the kiln?
I have two now. My small one is has an internal space of 12 inches, and my bigger one is 20 inches. And unlike ceramic kilns tend to be very tall because you can have several shelves of pottery. Glass kilns tend to be wide but very shallow, and that's to make sure that the glass is all evenly getting
it.
even temperature, because otherwise you get those stresses. They come
So
out.
we have another work here of yours. Is this a similar genesis, this plate?
So this is a little bit different. It's fused glass as well, but I've added some sandblasting to it. So there's a glass that I can buy that it's black glass and has a coating of a metallic oxide on the surface that gives it that iridescent. And I create masks, virus-shaped masks that protects that iridescent.
Okay.
And then I use a sandblaster and blast away the iridescent in the background, leaving just the viruses.
But it's also heated in a mold so it dips down, right?
Correct. Yes.
Anybody know what virus this is?
Looks like our friend Corona.
That's why you're wearing masks. It's because of this virus right here. I like that very much. So I'm going to commission one of these with our virus. Well, you can do that. You can request a specific one. So everything, Jane, that you make is some kind of glass, correct?
Correct.
All right. And this is
Yes.
pretty nice. So this is how you would display it. You would have it mounted on a piece of wood, a frame, basically, you can hang
Yes.
up, right?
Yes.
That's
But
nice.
that particular one could come off the frame and be used as a bowl.
Okay.
And you can eat your Cheerios out of it. Because hopefully I annealed it well. They're perfectly functional. And so you can, eat your Special K on your staff bowl.
I have a Zika virus plate, which I think Mark Martin gave to me many years ago. And I also have various pins. So you have earrings of a virus.
Yeah. That's coronavirus as
It's
well.
also
that I'm wearing.
corona. And so it's the same process. You just put a clip on it for, all right, okay.
Yes.
So Michele, here we have a couple of watercolors. That's one of your media, is watercolor, right?
Yeah, I mostly work in two different media. The main one is watercolor, which is standard, quite traditional technique. And the other one, this is ink. This is done on a surface called UPO, which is a polypropylene paper. And I can do several layers of ink with using a resist, which is kind of like a glue. And I can make whatever design I want, and then put the ink on it, let the ink do its thing, and it creates interesting organic patterns. And then I remove the resist, and you just see the white of the background. So we can talk a little bit more about like why I choose different media for different images. But yeah, I mean, all of my work is inspired by science, but I kind of do, I would say, a spectrum of like, if you look at this, this is a little bit more abstracted, the blood cells. I mean, it's not meant to look like, a photograph. The virus one here is maybe a little bit more realistic, but again, I mean, more like a textbook illustration type thing. And then sometimes I'm just doing something that's like much more on the abstract level. So just, depending what I'm trying to achieve with the painting.
It's, after all, it's art, right?
Exactly.
And as I said to Michele years ago, I said, this virus isn't quite right. She said, Vincent, it's art. It doesn't have to be right. And I always remember
I
that.
am not a scientific illustrator, and every so often people will approach me and want me to do some kind of an illustrating job for them, and I'm like, I'm not your girl, because, yeah, I don't really care how long the flagellum is if it looks better longer, I mean, look at Giacometti; those are not realistic legs. Picasso, really the eyes are not both on the same side, but, we get it.
In fact, if you know the Microbe TV logo, that was in part created by Michele, collaborating with
Peggy Muddles, the
Peggy
Vexed
Muddles,
Muddler on Twitter. Yeah.
And so some of you may know that I love plaque assays, right? What do you think she used as the focus for the microbe? A plaque assay. Brilliant. Absolutely brilliant. And I used that today, microbe.tv. So you have no scientific background whatsoever, is that correct? Just for high school biology?
That's pretty much the case. I mean, I feel like I've learned a lot over the past 10 years of creating art based on biology, but no, I can't keep up with you guys as far as really knowing what I'm talking about with these organisms.
Okay.
I'm mostly interested in bigger picture, really. I mean, the idea is just like, the first thing that I started painting was dividing cells. And I still paint those all the time because it's just basically life itself. And it's really just such a fascinating, endlessly interesting topic for an artist to be thinking about, like, what it is that makes us go, not only as human beings, but, the growing grass and the air, the water, everything around us. From there, I kind of got interested in the microbiome in the same kind of way as, like, here's this whole universe of stuff that's living in us and on us and in the air and in the water, and we really can't see it, but it's influencing us in so many ways. So that's kind of where, most of my inspiration comes from, is just thinking about those things and kind of trying to express that in art.
So you began with dividing cells, then added bacteria, and then where did viruses come in? Were they there in the beginning?
Well, I mean, I don't even know. This is probably going to get me in trouble, but I tend to lump the viruses and the bacteria together as like the,
They are microbes.
the small.
They are microbes. You're okay
yeah,
there.
Okay. But I also do a lot of neuroscience-related work. So, like this, as you can see, this is a pyramidal neuron, and over the course of the past couple of years, when we've had this little pandemic, obviously, as all of you, as all of us are, I was consuming a whole lot of news about the pandemic. I was consuming a lot of science communication that was trying to tell me what was going on. And as the pandemic unfolded, I had this really strong feeling that, like, just the science wasn't really cutting it as far as, like, getting across the messages of what was going on with the pandemic and how it was affecting society and so on. So I started using more literature, and I've been doing some pieces that involve literary poems or quotations about memory or about consciousness, and I'm using those words in here. Just trying to sort of bring together not just the science, but also, like, including a little bit more of the humanities, because I think it's really, really important to communicate using everything we've got. Not just trying to, like, hit people over the head with facts, but the art is a way to reach people, literature is a way to reach people, lots of different ways that we can try to communicate the whole picture.
Yeah, I was just talking to someone about that, and a painting or some kind of an art depicting a disaster is often more impactful than talking about it for an hour, right? So artists have a big role to play. Now, do you have any kind of art training?
Not really.
Okay, you picked this up on your own. Great.
Yeah.
Well, that's very good. And you have, what is geology?
Geology and paleontology, a lot of zoology,
But
no art, no formal art.
no formal
training.
art training, yet your subjects are microbes, viruses, bacteria. Where did that come from?
Well, when I started in glass, I started with just regular decorative, ordinary stuff. Did craft fairs, and I did okay. But then just for fun, because I was interested in science, I started making science-themed items,
Okay.
and they sold right away. And I thought, oh, there's a market there. So I started making insects and fish and a whole variety of stuff. And then people would walk into my craft fair booth and said, well, hey, can you make some E. coli? Can you make a dengue fever? And I'm like, well, sure. That sounds great. So I started learning about microbiology. So I do a lot of looking on Google, looking at images, and I try to see what's important about each microbe. What are people talking about? What parts do I need to include? For instance, flu, they talk, the H1N1, the H's and N's. Okay, there's something there, so I better, depict those. So I always make sure in my flu to have the, to distinguish between those. And the 8 segments of.
The 8 segments
yeah.
are very important for the shuffling part.
You see, this is where Jane and I are like, we're parting company on this because I'm like, 6 segments, 8 segments, whatever.
I like that.
So how
Yeah,
did
so-
you get into glass? That's
I-
what intrigues me, because glass is not easy. You have to want to do glass.
Well, it's just a skill like anything else. It's
Yeah.
not as hard as it looks. People are intimidated because, oh, you might get cut. You might burn yourself. Well, I get cut all the time. I've got a lot of Band-Aids in the house.
And you cook, so you burn yourself all the time.
Yes. So I'd always been intrigued, particularly with stained glass, and I had a friend who did it, and she invited me into her basement one day, and I made my first stained glass panel, and then I was off and running. So that's how I got into it. Yeah, and it's just learning and practice.
Yeah.
Yeah.
Well, that's what science is. It's all about learning and practice.
Exactly. And there's a lot of science in glass art. For fused glass, you can take 2 colors and fuse them together, and at the joint between those 2 colors, there'll be a chemical reaction, and you'll get a different color between them. Now, I like doing that. If I need a cell wall, for instance, I can choose 2 colors that I know will react with each other, and I'll magically get a nice line around.
Is that
from
what happened on that plate that Vincent was holding up earlier? That one with the Staph aureus with the beautiful color changes?
So in that case, no. In that case, so that I started with a green circle, and then I put a slightly smaller dark blue circle on top of that, and then a light blue circle on top of that. And so that it fuses together. And then the little sprinkles for the
The
frit, right.
frit.
For the DNA and the cytoplasmic stuff.
I'm just curious, how long would it take you to make this? How many hours? Well, you don't keep track, maybe.
We don't talk about how long it takes us to make things.
Okay, artists don't do that? Okay.
Several hours, I'll just say that. That particular one, several hours. And that's not particularly complicated.
Do you ever
So-
get outcomes you haven't planned?
All the time. All the time. And sometimes it's even better than what I was planning. It's like, ooh, that's cool. I'm gonna learn what's happening here and use it again.
Right, right.
Over time, I've learned the material science of it, so I have fewer surprises because I've learned what'll happen if you use this color or that color, what happens if I have the kiln at this temperature versus that temperature. I recently did an experiment with salt, table salt. I thought, what would happen if I fused it as an inclusion, they call it. So I had a base layer of glass, put some salt on top of that, and then capped it with another piece of glass. And the salt melted into a puddle because the melting temperature of sodium chloride is less than that 1,500 degrees. So it melted, and then when it cooled, it recrystallized into these gorgeous interlocking flat crystals, and it was very cool. I had no idea when I started what was going to happen, so that was a really cool experiment.
And Michele, same thing, you get outcomes you don't plan often?
Absolutely. Yeah, I mean, I'm sure we've all heard that, the saying that doing the same thing over and over again and expecting a different result is the definition of insanity, but it's also the definition of watercolor. If I take a brush and I'm dropping watercolor, into an already wet field, I'll do that 10 times and it'll be different every time. And yeah, I mean, that's part of the beauty of it, is just never knowing exactly what you're going to get.
Do you get inspiration from other artists at all?
Oh, absolutely, get inspiration from other artists. That's one of the things that's been tough, like over the past couple of years, not being able to go out and see other people's art. But yeah, I mean, there's a huge spectrum of people who are working with science from, I mean, look at Johnny over here, who is like, we're both completely in awe of his ability to just sit there and draw like that and just off the cuff be able to create people's, whatever organism they're working on and do it in such a unique way. To way the other end of the spectrum, you have somebody like Olafur Eliasson, who is, really famous Danish Icelandic artist. One of the things that he does is take chunks of glacier and take them to climate meetings. And I mean, this may seem a little bit like a stupid thing to do because obviously the glaciers are melting already, but it makes a point to people about what you're losing. Because I mean, his work really, really resonates with me, even though it's very different, in technique, obviously, from what I do. But if you think about the information that we get from a glacier, the little bubbles inside a glacier tell us so much about what was going on in the atmosphere, hundreds or thousands of years ago. When you have the glacier, when the glacier melts, all that information's gone. It's literally just a drop in the ocean. So, I mean, to me, that work is really beautiful and moving, and it's also just very, it makes you think.
What do you got there, Michael?
I have some buttons from Johnny.
Cool.
So I'll give them to the ladies to hold up. These are absolutely incredible. You can see one, we have a bandit bacterium, and then the other is probably a sleeping helicobacter in our stomachs. I mean, these are just incredible. I don't know what the bandit is. It's got a piece of cheese, and it looks like it's got a CRISPR sequence. It's a pizza, yeah. The standard graduate student diet, pizza.
So, Jane, are you inspired by any particular artists?
I'm certainly inspired by artists, nobody in particular. I do enjoy seeing, the wide spectrum of how people, what colors they choose and how they do, I'm inspired by Michele's work. I like to see, how did she do that virus? I tend to get really caught up in putting in all the details. and I like going to Michele to see, it's beautiful and you know what it is without a whole lot of details. So I learn from that. Yeah.
I forgot to mention, Michele, besides watercolors, you have clothing as well, right?
Fabrics.
Well, I don't really make those myself. So, I mean, yes, I love them. I have ties and silk scarves made from from some of my paintings. But yeah, I don't actually make the fabric myself. So anyway.
So you send the image to a company and they print it on fabric, right?
Yes.
So you have ties and scarves, right?
Right.
And maybe soon bow ties.
Maybe soon bow ties.
Maybe we're gonna
Maybe
see
socks.
Yeah. I
Maybe
mean,
socks.
Would you
Watch
guys
this
wear
space
socks? Because yes, I obviously the general trend is for people to be wearing ties less. And I think, not that many people wear scarves. So I am kind of, thinking about maybe what could be next in that. Socks are a great idea. Umbrellas. Everybody needs umbrellas.
Mm-hmm.
Yeah, look at ASM's umbrella. It's very boring. It's just black. You can see it back there.
You would
Ray's
seriously
yelling
want
at
an
me.
umbrella with microbes all over it. Absolutely.
Well, they already have microbes all over them. You just can't see them.
So over the years, Jane, have you seen an increased interest in science art since you started?
Absolutely. Both from the science world and from the general public. Mm-hmm. It's really cool. When I was in school, 1980s, scientific art was very, clinical, black and white and simplistic. And I've really enjoyed seeing the scientific world embrace more color. I think it makes it more accessible to people if you make it interesting, if you make it pretty. And certainly more and more people come to my work and say, "Oh, this is cool. I studied that in school," even if they're not science. They see a planarian. I make a lot of planarians, and they remember when they did that experiment in high school, yeah.
I think that making it accessible really helps, right? If they see something, oh, I know what that is, and plus
Yes.
it's beautiful. It's a piece of art, and
Yes.
it connects, right?
Yes.
More than a PowerPoint presentation.
And glass is really great for the color, and I think that really opens up the doors for people. If I make something yucky, I'll make a tapeworm, but it's a pretty color, and I might make, it look like some personality, almost eyes, and people will go and say, "Oh, I didn't know tapeworms were pretty." And so it keeps that door open to communication about tapeworms, because they're not immediately shutting down, "That's gross, I don't want to see it."
Yeah.
It's pretty, so that opens up the communication about difficult subjects. Yeah.
Yeah, I mean, I'm just going to say, all of you guys here probably know this, but the outside world doesn't necessarily know it. But if you magnify anything enough, it gets really good looking. So you can, I had a beautiful painting that I sold yesterday of black mold, which, you would be really alarmed if you saw it in your bathroom, but under the microscope, it's gorgeous.
Coronavirus is a beautiful virus. I just, those long spike proteins, it's just lovely. And all viruses, I love they're so geometric, and it lends it well to art and doing really beautiful things with them.
Michele, have you also seen an increase in interest over the years?
Oh, definitely. I see more people doing science-inspired work, and more interest. And again, like I said, there's a big range. I mean, there are people who are, professional illustrators, obviously, but who, doing a lot of really interesting work. One of the things that I like to plug is going from is the role of art can take something from data visualization to what I like to call data visceralization, where you're really just getting it in the gut, where it's putting across a message that you feel more than you absorb mentally. So that's something that I think that it's going to be incredibly hard to measure, but it can be powerful.
Even our friends at the National Institute of Allergy and Infectious Disease, when I was coming up, EMs were dull. They were black and white and flat. Now, NIAID in its public image database is putting up more and more colorized EMs. And they really tell the story of how the microbe is interacting with the host, and you can get the sense of what the microbe is doing, and more importantly, the action, how they're doing it. And that is what is conveyed in your art. My dad was an artist, and he drug us to art shows as tiny tots, and we didn't much like art shows, but he taught us that what art had to do was move. It couldn't be flat and static. And if you just look at some of these images, you can see the movement of the art. And it's absolutely stunning that you're able to capture that this is life. And that is, I think, your inspiration in everything from the glass to the paintings to some of the incredible fabrics that you are making and displaying over in your booths.
Yeah. Well, I'm just picking up on Vincent's question too about and both of your comments here. I think that the advances in scientific imaging have really just made it so much more appealing and accessible to people to work with this stuff artistically. I mean, X number of years ago, you just couldn't see the things under the microscope that you can now. And they're incredible. And, the things that we're learning, that really can, pull you in intellectually, but also the beauty appeals aesthetically.
So when you decide, maybe let's start with this. How do you figure out what you're going to do next? So Jane, when you wake up in the morning, how do you know what you're going to make? A dengue or a Zika or a staphylococcus? What drives you?
Well, I get a lot of special requests, commissions, so that's a big driver. In the process of doing a commission, I will be testing out new techniques and stuff, so that'll take me down a path. So that's one driving the requests. Sometimes something in the news will catch me. Some newly discovered crustacean in the ocean that it's like, oh, that's cool. That would look good in glass. My family gives me ideas. I'm thinking about what's going to sell at the ASM Microbe meetings. That drives me. I should have brought more gonorrhea. It turns out that's a hot
Gonorrhea
seller.
always sells.
Cholera. I am sold out of cholera. That was a hot seller.
All the fungi people are like, where are the fungi?
That's great. How about you, Michele? What drives you?
Yeah, same thing. It's kind of a combination of practical and artistic. So it's a combination of where I'm going to be showing next. And also, but I definitely work in series. So I'll get started on something and then I'll just keep doing versions of it and it'll get better and develop in different ways. But I mean, I think that's pretty typical for most artists. So a lot of, like, when I'm not busy working on work for a particular show, I'll just be like trying to develop some, new sort of thing, new image or new technique, and just be working on that. But I'm also just going to pick up on your point of, like, you didn't much like going to art shows.
I was 5.
I know. Okay, but a lot of people don't much like going to art shows. It can still be a really intimidating thing. And it's very difficult getting people to go to art shows. So I'm partly putting this out here as a plug to get the artists to go to your science meetings. Because this is
Good
nice.
idea.
Yeah, I mean, honestly, it's great for us, but it's also good for, the attendees to, see what we're doing and talk to us and think about how art and science can work together. And I actually was for a short period of time artist in residence at a fruit fly lab, for example. Mm-hmm. So your lab can have an artist in residence. I mean, you can do these things.
What does it mean to be an artist in residence? What do you do?
So I got a grant.
Okay.
And I found a lab that was doing really interesting work on evolution using fruit flies. And basically just pitched it as a project where I would work with them, observe what they were doing. So I came into the lab every day, and I actually learned to cut up fruit flies.
Okay.
So
Maybe dissect is
dissecting fruit flies, that's the word. And basically just really learn what they were doing. And one of the things that really hit home to me was, like, how much of it was still one at a time by hand, which I was not expecting. So there was a split in the lab between the people who were doing the real sort of, like, I would say maybe old-fashioned, bench work, where they're dissecting flies and looking at them under the scope. And then there are people who are just working with the data. So, it was definitely a great learning experience for me to see, what that was like, and how that feed into my work. And also just the idea that, you can take something as tiny as a fruit fly and look at one single part of it. I mean, the lab that I was working in, they were working with one fly that lives in one island, and you can, based on that, extrapolate about evolution, which is one of the biggest topics ever, you know, I mean, so really interesting to me to see that through line and to try to reflect that also in my work.
Did some of those works end up being sold from this?
Oh, yeah.
Yeah. Okay. Sorry.
See, I always thought that Picasso was reincarnated as a fruit fly geneticist who created the antennapedia mutation, where they have this foot coming out in the wrong spot.
Yeah, that's good. That's good. You haven't done that sort of thing, right?
I have not done a residency, but I've done several collaborations with scientists, which I really, really enjoy. In my booth right now, I have this big piece featuring cyanophages attacking microcystis, a cyanobacteria. And that was made for a science conference in California where they paired up artists with scientists. The conference was focusing on the science of the Sacramento Delta. Maybe I already said that. So I worked with a scientist who taught me all about microcystis, about cyanophages, how it works, you know, why it's important. It's one of the few controls of microcystis, and the Sacramento Delta has, like many places, has a lot of trouble with harmful algal blooms. And this is really, really satisfying for me because not only am I learning some cool science, but it drives my art because I'm thinking, how am I going to depict this? You know, microcystis has a slime layer, I think it's called a slime layer, around it. It's like, how can I show that? So I'm looking for the right glass, and I'm doing tests to see if I can make something that looks slimy. How am I going to show the reproduction of the phages inside the cell? So it's driving my art as well as teaching me new science. And I work a lot with labs who want to present a present to students who are graduating or people who are leaving. So they will work with me, and I'll make pieces featuring the research of that student or the person who's leaving. And again, it's very satisfying, and going back and forth and learning about it. And I'm always tickled because they seem so pleased that an artist is interested in their science. And I'm so tickled because they're interested in my art. So it's really a win-win.
I think this is very interesting, the idea that you both have to figure out how to depict something. I never thought of that approach in glass and also in watercolors.
Yeah.
That's specific to art, but yeah, I mean, it's not a photograph, right? You have to depict it in some way.
And if the art, you want the art to tell a story, and so that means you can't necessarily put everything in, or it'll become too cluttered and you'll lose the message.
Right.
So
Right.
I'm always looking at what are the important parts? What do I need to put in there so that it tells the story without getting lost among all this other stuff? Yeah, and that's the challenge frequently.
I call this essence of pig. The idea is that you can create like an incredible painting of a pig with like every detail right, but you can also do a round circle and then another round circle and two round circles inside that and some little eyes, and you have essence of pig.
Not Mickey Mouse?
You've put across pigness, and everybody can recognize that as a pig. And I actually have like kind of ongoing beef with Michael Eisen online about DNA going the wrong way. Who cares? DNA doesn't really look like that anyway. You can tell what it's supposed to be.
Yeah.
And I think this is where our science art can be better at communicating than actually a photograph, because we've gotten down to the actual message, communicating what that story is. Whereas a photograph, it's kind of hard to see. Scientists will send me their photograph, and they say, can you make something with that? And frequently, my first question is, what is it, and what am I seeing? What are the parts? And I'll even ask them to, take your crayon, and don't worry about artistic ability, and just do a line drawing of that photograph. So that helps me see what they're seeing in that photograph.
All right, we have run out of time, so let's wrap up. That's a special episode of TWiV. You can find the show notes at microbe.tv/TWiV. Questions, comments, TWiV at microbe.tv. And if you enjoy what we do, science communication this way, please support us, microbe.tv/contribute. Our guests today, science artists from Trilobite Software, Jane Hartman. Thank you for joining us. Appreciate it. And for explaining your process. I think it's really useful.
Thank you so much.
And people can find your booth right next to us here, right?
1514, just around the corner.
Please go check it out. It's beautiful stuff. And where can they find you online?
So you can go to Etsy and search for Trilobite Glassworks.
Thank you. And from Artologica, Michele Banks. Thank you, Michele.
Thanks so much for having me. This is great. And yeah, you can find me on Etsy also at artologica.etsy.com, or just Google Artologica. I'm out there.
And so I have many of works of both of you. And a few weeks ago, Michele put an Instagram picture of a new drawing of neurons, all in blue. And I said, I need that. Don't sell it. And she shipped it to me. It's now hanging in the incubator in New York City. So I really appreciate it. In fact, our recording studio in New York, the incubator, is full of science art. And I think this is the best showcase we can have. We can have videos and podcasts, and we can have science art as well. And it's all working together. So thank you both. And thank you, Michael Schmidt from the Medical University of South Carolina, for joining us today.
Thank you. I really enjoyed it.
Now, you didn't like art shows as a kid, but now you probably do, right?
I do. Yeah, my father's artistic love rubbed off on both my brothers and I, and we were grumpy as 5-year-olds, but we have a sophisticated eye now, and we know good art and we know bad art, and there's a lot of bad art.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and in particular ASM for having us here today, and Ray Ortega for the production. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.